that requires energy. So no energy. This is all called passive transport. Okay, we got some flavors of passive transport. What do we have? Osmosis. Osmosis? What is osmosis? Yeah, diffusion of water. Say again? Just regular old diffusion. And facilitated diffusion. What makes it facilitated? There's some kind of membrane protein housing. Okay. All of these passive transport mechanisms, do they make the inside of the cell more like the outside of the cell or less like the outside of the cell? More. Yeah, then they get more signal. Okay, but what if I want the inside of the cell to be different than the outside of the cell? Yeah, I've got to use active transport, so energy required. Active transport. What makes it active? Yeah, it uses ATP. What uses ATP? Yeah, some kind of membrane protein uses ATP. A membrane protein, we often call it a pump, uses ATP to pump something across the membrane. Okay. This is good. Active transport is good if we've got little small molecules that we need to move across the membrane, like glucose molecules or ions or something like that. Then active transport is great. But what if I want to move something really big, like a big protein? Yeah, then I have to use what we call bulk transport. And there's bulk transport going into the cell and bulk transport going out of the cell. So if you are transporting a large something into the cell, what happens is you've got your cell and your thing that you want to take in. Maybe this is a protein, maybe this is a bacterium, maybe this is some little cluster of tasty things. Who knows what that is? What happens to the cell? How does it get this thing inside the cell? Yeah, the membrane engulfs it.
Is that only white blood cells or is that other cells?
It's other cells as well, but often for smaller things. So this could, like white blood cells take in like whole bacteria, but for example, your nerve cells will take in like single proteins into tiny little vesicles. And what you wind up with is the thing that you're taking in has been taken into a little new vacuole or a little vesicle.
So this is a typical science classroom. You can see a classroom setting in the front and a lab setting in the back, which is how we normally apply for lab spaces.
Oh yeah, so this requires energy. Why does this require energy? What does the energy go into? It's not powering a pump.
It's going from a low concentration to a high concentration.
Yeah, it's not even really going to a different concentration because it's not contained in a little thing. But how do we actually move the cell?
It takes energy to change the structure.
It does. It takes energy to change the shape of the cell. So the cell actually has to grow its cytoskeleton out around this thing to move around it. Otherwise, it can't move and take it in. So it's actually the cytoskeleton regrowing that requires energy. Really good option. In the cytosis, we're going to engulf particle in membrane, and now it's inside the cell in a vacuole or vesicle. And from there, we could actually turn this vacuole into a lysosome by adding some digested proteins to it that digest the inside. Okay, sometimes cells want to take large things in, but sometimes they actually want to spew large things out. Either a signaling molecule, maybe they need to like spew out some proteins to signal to other cells, or maybe they have like large blobs of waste that they need to get rid of. So how do you get something out of the cell that's big? Yeah, exocytosis. So roots to know, endo means in, exo means out. How about site? No, lyse is cut, site is cell, cyto is cell. So we're going out of the cell. And this is just the reverse. So we have our thing that we want to get rid of, and we want to contain it in some little vesicle. The vesicle moves to the membrane, fuses with the membrane, and then it expels its contents. And so since all these membranes are made of the same stuff, they can all just kind of fuse together. So expels contents.
Yeah. I was that good. Yeah.
By fusing. And again, this takes energy because we have to move those vesicles around, and we have to regrow our cytoskeleton in order to do that. And that requires it. Yeah.
About facilitating.
Yeah.
So, are the proteins on the membrane just like there all the time and then they still like uses it when it needs to, but it doesn't require energy?
Yes. So actually making the proteins requires energy, but once they're there, they don't require any further energy to work. That's why it's passive. But the cells can actually put them onto the membrane or take them off of the membrane. And some of them are like what are we call gated, which means that they're like closed, but then if something binds to them, they open up. So the sodium channels in your nerve cells work like that. You have gated sodium channels, and so they're closed all the time, but if something binds to them, then they open up, and that doesn't require any energy, and then stuff can flow across. Yeah.
Can you repeat what you mean by fusing?
So the vesicle is made of the same kind of membrane as the cell membrane. So when the two like touch together, they just like loop into one sheet. So the like vesicle touches the surface, and then it like becomes part of the cell membrane, and it like dumps its contents on the outside. And that's like it for transport, which means that that's kind of it for cells. That's it. That's all I need to really. Yeah.
Will we like take over like kind of like go see stuff in there?
I'm not that concerned about it. It's just another form of bulk transport, just with water.
Coming out here.
Yeah. Yeah. Let's look at our demo. Sorry. I'm gonna just like hit the board here real quick. Yeah, that's what it says. All stuff on the board. Okay. Okay, let's go look at our demo. Someone want to be a camera person?
I'll be the camera
You want to be a camera person?
Yes.
Okay. Here, I'm gonna move over here. These are our demo beakers. Alright, let's start with this one because this one is the one that is the most interesting. So, what do you think? What can move in and out of the cell? How did it start?
Wasn't it yellow because of the iodine on the outside?
Yeah, previously it was yellow on the outside because of the iodine.
And then-
starch and glucose.
Starch and glucose were on the inside. So, how about the starch? Did the starch cross the membrane?
No.
How do you know?
It's not yellow. It's
not
not
blue
blue on the outside.
The outside
It's not blue
didn't
on the
form any
outside,
color,
it's blue on
only
the
the
inside.
inside.
Right, only the inside is blue. So the iodine binds with the starch and turns it blue, and now all of the blueness is on the inside, so the starch is on the inside. How about the iodine? Can the iodine cross the membrane?
Yeah. Is that why it's like all gone? Because it binded with the.
Yeah, it actually, when it hits the starch, it binds with it pretty permanently, so it's not yellow on the outside anymore.
Okay.
How about
it,
the-
like waterproof or because how would it be like permeable?
It is not waterproof. Water can flow through it, it just doesn't flow through it very quickly.
Okay.
So that's why it took overnight for it to kind of happen.
It's starch proof.
It's starch proof,
Okay.
yeah, it's starch proof.
Is it
How about the glucose?
We can test.
We can drink the water.
No. Gross. Okay, I actually think I only have one urinalysis strip left. I had exactly enough. So where do I want to test?
Can you
Can become
the water
Yeah.
The water that was inside or the water that was outside?
outside?
Yeah, the water that was outside. Because the glucose started on the inside. So we'll dip that in there, and it takes it a second, so we'll let it develop. Here, hold, go away.
It's really blue.
So we'll let that work. I'm not sure whether this one will have worked much or not, but we can find out. So this was our isotonic. So this is what the cells looked like when we started. This one is our hypotonic. So let's see. Are they visibly different from each other?
Yeah.
What do you think? This one is
Cloudy.
Has a starch
cloudier.
in it.
Yeah, it had the starch in it. Do they look different in size? Not really.
Yeah.
Yeah, I think maybe this didn't work as well as I was hoping that it would. Maybe I should have tested it beforehand. How about this guy?
Just water. They're the same.
Yeah, they seem the same.
They seem kind of the same.
Yeah.
Dang. This works really well with eggs.
Does that have more?
Which one's supposed to have?
Yeah,
It's
well
supposed
this
to
one
be
this one should
less.
have
Less.
less. This one should have less. It should diffuse out to the starch
I
side.
mean, it has
This
a-
one is isotonic, so it should be like our control. And how about this one?
That should be more.
Should be more. We could actually see if there is slightly more or less. We've got some graduated cylinders. So let's get the ones that are like 25. Let's go. What do we got?
Hypotonic, so the water moved in to kind of do better starch than
More water on the outside.
Okay, this one was hypertonic. It was in a hypertonic solution. This one is our control isotonic. Gross, you're gonna get my gross finger on film. And this one was hypotonic.
So the one that should have.
So this one, what should have happened to this one?
Should have more.
Should have more. Let's see if it has more. Well, this one definitely looks like it has less, right? This one's got about 15.
This one's thick.
This one and this one, the tubes aren't the same, but they're
Yeah.
both this one's a little more than 20, this one's a little less than 20. So it didn't really diffuse in. Maybe it doesn't have very much room to stretch. Maybe it can actually push
Maybe
the.
it's too slow.
Yeah, maybe it's too slow, or maybe the like the membrane provides too much resistance. It like can't like fill up very well. It's not like a balloon. It's not very stretchy. But anyway, that one is definitely less, right? So that's good.
Wait, so the hypotonic solution has more concentration on the outside?
The
Hypo.
hypo has less concentration on the outside. So it should always go towards the more concentrated thing. And the more concentrated one is the hypertonic one. Okay. So that's kind of cool, right?
Cool.
All right, that's it, demo. So our cells are actually it's just a size separation in the dialysis tubing. It actually doesn't really have a polarity element to it. It's kind of just a non-charged substance. I don't know what dialysis tubing is made of. Okay, cool. Thank you. All right, so we're going to talk about something completely different for the rest of class. Any other questions on cells or organs? Oh, wait, I forgot. What we're going to do is not completely different. We're going to talk about cells. We're going to go through that inner lines of a cell video and see what we can see now that we know what the organelles are. And then we'll talk about something completely different. I have two videos. This one I just kind of like. This is endocytosis in action. These are immune cells. These are white blood cells that are just chowing down on bacteria. Which is kind of neat. Here we go. Exciting music. Anyway, that's all sped up, but that is what your white blood cells, some of your specialized white blood cells do.
Wait, the video sped up or like the actual process in the body is way faster?
The video is a little sped up.
Compared to the process?
It is pretty fast, it's just not quite that fast. I don't think.
So does cough medicine like act like white blood cells, or is it usually?
I think cough medicine is suppressing your nervous system's need to like cough. You have like several different types of immune defense. So one of them is like eating bacteria. One of them is like marking bacteria for destruction with antibodies, which is what vaccines help promote antibody production. And some of it is like really physical, like physically expelling the bacteria through coughing. But yeah, we actually don't. None of the medicines that you take act like white blood cells. They either just like kill the bacteria outright or make your white blood cells better at their job, but we don't actually take in anything that can eat the cells. Yeah, until the days of nanorobots that help us with our cells. Okay, I'm gonna slow down. This is the inner life of a cell video. I'm gonna slow it down. I kinda like it at 0.5, but it was being real laggy at 0.5 yesterday, so we'll see how it goes. Okay, so, here we go. Where are we in the body right now? Yeah, we're inside a vein. So those little red things that are flying past, red blood cells. And what we're looking at is that blue thing that's crawling along the top. What's that guy? We just saw it in the other video. It's a white blood cell. It's called a macrophage. So it's patrolling the veins right now. And we're going to zoom in on that cell. Okay, now where we are, this is the surface of the white blood cell. This is the surface of the cells that make up the vein. And so these two cells are actually, as this white blood cell crosses, these guys are talking to each other through proteins. I know, crazy. So we're way slowed down. So these little proteins are talking to each other, and they send some kind of message. I don't know 100% of what's happening in this video. It is all depicting something specific. Okay, now we're inside the cell. No, wait, no, we're on cell surface. This is the cell membrane. The blue stuff is the cell membrane. We've got proteins floating around in it. Protein floating, and cells communicating with each other. And now we're inside the cell. We're looking up at the membrane. Okay. And then we got, I don't know what this is. It looks cool though. I don't think so. Because it doesn't look the same. What's this? This scaffolding we're looking at. This is the cytoskeleton. And so it doesn't look the same as those triangular things. I don't know what those triangular things are. They seem to be in the membrane. So here's the cytoskeleton. And here's a piece of cytoskeleton being assembled. This is a microfilament. And so the cell can kind of move around by changing the cytoskeleton. Here's another piece of microfilament being disassembled. So the protein hooks on and then it breaks apart. This is the other type of cytoskeleton. This is a microtubule being assembled. Each of those little building blocks is a protein. And then they disassemble.
And the process is super fast.
Super fast, yeah. Did you guys read that blog post?
Yeah.
Yeah.
And,
Yeah.
But then, like, for white blood cells, the way they move is not as fast.
Oh, inside the cell, stuff is super, super, super fast. It's just that the cell itself can't quite like zip around like that.
Oh, okay.
But the molecules inside the cell are super, super, super fast. So,
Okay.
you guys remember how fast this guy is going?
Sprinting.
He's sprinting at 100 steps per second. It looks like it's just trudging along. And it doesn't show it, but every time it takes a step, it's using an ATP.
Oh.
Yeah. You use a lot of ATP. And what's that big blue thing he's carrying along? What is it? It's not a vacuole, it's smaller than that. It's a vesicle. It's a little bitty vesicle. Alright, what's that in the middle there? That's the nucleus. And yeah, that looks like a mitochondria, this sort of water bearing thing.
What is it? G. This is a G.
This Golgi, this is the Golgi body over here. We'll see a better look at this Golgi body in the future. We're not going to go inside a mitochondria in this video, but it's got kind of this double membrane. So that's how you can tell what it is. What else did mitochondria have that make them special?
Their own DNA.
Their own DNA. Alright. Right now, we're at the surface of an organelle, and something is coming out of it. Where are we? Where we are? We're at the surface of the nucleus, and what are those little things that are coming out? Those are RNA. Yeah, those are RNA. These little RNA strands come out. Yeah, they're gonna bind to ribosomes, so they kind of loop up. I don't really know why they're doing that. I don't know that always happens, but here comes a ribosome, it's that green thing. There's half of it. And here's the other half. Coupling. Now what's going on? What are you guys saying?
It's like instructions.
Yes, yes. This RNA strand, this is instructions for protein, and this is the new protein that's being made. Protein. Here's two proteins that are forming a dimer for some reason. What's that big guy again? That's mitochondria again. Okay, here's a ribosome. It's coming down to the surface of a membrane. Where are we? The rough ER. This is the surface of the rough ER. So the rough ER has this little pore in it. The ribosome lands on it and makes the protein on the inside. See it underneath the membrane? New protein inside the ER. And the ribosomes actually pop on and pop off. And what we're seeing here is a really good example of, it's not exocytosis because we're not outside the cell, but see those little vesicles being formed? The little vesicles are being formed on the surface of the ER. We're getting lively. It would be exocytosis if it was leaving the cell, but it's actually just from organelle to organelle. We're still inside the cell. We've got another little vesicle going.
100 ATPs per second.
Yeah. You use a lot of ATP. What's this guy? This is Golgi. And you can see there's like vesicles coming in and merging with it at the bottom and then blobbing off at the top. That happens all the time. There's like proteins going in there being changed, being modified. And now this is exocytosis. The blue things are being expelled from the membrane. And all these orange guys are membrane proteins. Want to go again? Yeah, we're now at the top of the cell. Whoa! What happened there? You can do it. And now we've got these membrane proteins coming together. I'm not sure what this like rippling in the membrane is, but it's cool. I'm sure it's representing something that's happening. These are membrane proteins. We're at the surface of the cell. Right now we're in between those two cells again. So top is the vein, bottom is the white blood cell. And these guys are gonna actually change shape, hopefully. And they're gonna hook on to these proteins up here. So what's happening is the blood vessel is sending a message to the white blood cell. It's saying, there's a problem, I need you to come in and invade. I need you to find what the problem is. So the white blood cell is rolling along the surface. It gets the message to stop, and it stops. And then it's going to change its behavior. It's going to like squish out. And it's going to actually slide in between two cells. And it's going to go find some bacteria, or whatever the problem is, and fix it. Yay! So the reason that if you have like an injury and it gets like swollen, part of the reason it gets swollen is actually white blood cells squeezing into the area and looking for bacteria.
So like when you squeeze out, it can be really, really thin.
Yeah, they like flatten and then just in between cells. Alright, neat. Alright, now for something completely different. Totally different. How much time do we have? 10 minutes. I'm gonna give you guys some context for life on Earth. Big picture context. We'll go up one light. No, I like no lights because I like these slides. Okay. So you don't need to worry about taking notes for this. So, context, the universe started about 12.5 billion years ago with something that we call the Big Bang, which is where prior to that there was no universe, and then the Big Bang happened, and now there's a universe. So that's pretty good. That was a good thing to happen. We actually still have the light from the Big Bang is still present around us today in radio waves in the sky. So if you point a telescope up at the sky, a radio telescope, there's this background radiation everywhere in the universe of radio waves that's still the light and the sound of the Big Bang. Which is pretty neat. So at first there was just a bunch of energy and some matter, and it was kind of evenly dispersed. But eventually it made some atoms, and that was a pretty good step. And fortunately, those atoms weren't completely evenly distributed everywhere. There was some clumpiness. So where there was some clumpiness, gravity started to pull things together. Eventually it pulled a lot of atoms together. So many atoms together that it got really, really hot, and it started a nuclear fusion process. And what did it get? It was something like that. Huge ball of atoms. Now they're fusing things nuclearly. A star. Yeah, we got stars. That was good. It was a good step for the universe. These are some stars. Originally stars, they only had one kind of atom, which was hydrogen. And the hydrogen came together in a process called fusion, where two hydrogens sort of smooshed together and made more complex elements. So first they make helium, and then they run out of hydrogen, and so the helium starts to fuse, and you can get lithium and beryllium and all of the way up the periodic table. And every time you try to make a new element, the star has to get denser and hotter to do more energy-intensive fusion reaction. So the star gets like denser and hotter, denser and hotter, denser and hotter. And eventually, a star, there's a star, that's our star. Our star is a young star, though our star wasn't even born yet. Eventually, you get iron starting to accumulate in the middle of the star. And iron doesn't like to participate in fusion reactions. So, instead of getting a little denser and a little hotter and fusing the iron won't fuse, so it has to get, it collapses. The whole star just collapses in on itself. The center of the star collapses. It gets like super dense, super hot, and all of the outside part falls into the middle. It all falls into the middle, and then it hits the middle, and it explodes outward, bounces out in a giant explosion called a supernova.
Isn't that what's going to happen to our star or our sun?
It is not. Our sun is not big enough and not hot enough. So
But
it's,
it will eventually happen?
it'll never happen. We don't have enough mass in our sun. So our sun, it will expand at some point and become a sort of a, I can't remember what the name of it, not a red giant, but something slightly smaller than a red giant. And then it'll sort of collapse, but it won't supernova. But when it happens, it's kind of done for a second. It's gonna like fizzle out. It's not gonna explode. But this is what's left when a star explodes. All of this clouds of stuff. It's called a nebula. And this is great because now, in addition to hydrogen and helium, we have all of the other elements. In fact, all of the elements, because when the star supernovas, there's so much energy that iron will fuse, and you get all of the elements up through uranium. And so now we have this cloud of dust that also contains carbon, and also contains oxygen, and also contains nitrogen, and all this stuff that we're made of, which is good. So eventually, a nebula like this, gravity is going to pull it back together, and it's going to reform a new star. Here's another nebula. Nebulas are star birthplaces. So all of this space dust will coalesce, come together even more, get so hot and so bright that it makes a new star. And surrounding this star is a disk of dust, and this dust will all come together and make planets. And so you get a disk of planets instead of a disk of dust. And that's pretty good, because we need one of those planets to live on. And so 4.5 billion years ago, we got one of those planets to live on. This is the sun igniting. I think this is not quite accurate, this scale. This is about 4.5 billion years ago. We got the sun, and we got the earth. And then a lot of things happened, and I like this graphic because it's pretty, but we're going to look at it in a little bit more depth. Okay, so for context, we're going to say, like, what if we took all of the entire history of time and we compressed it into a single calendar year? So like right now, this second is New Year's. It's like midnight on New Year's. And all of history occurred over the past year since last New Year's. Okay, what would that look like? These are the pre-December dates. Okay, so Big Bang, January 1st. Our galaxy didn't show up for another 5 months. We're here in May. Our solar system, our sun didn't appear until September 9th, so this is happy birthday, sun. It didn't show up for 9 months because we had to have a whole cycle of a star being born, living its entire life cycle, exploding, and then coming back together into a new sun in order to have our sun. Earth formed shortly after the sun. And life on Earth showed up almost as soon as conditions on Earth were stable enough to allow it to, which is pretty neat and kind of a good sign that maybe life is common in the universe, because it showed up on Earth so quickly. It wasn't like it took another billion years for life to show up. It came about pretty much as soon as the surface of the Earth stopped being magma, which is cool. These are the oldest rocks that we know of. Were formed on October 2nd. Oldest fossils, October 9th. Invention of sex on November 1st. So that was, I guess, an exciting time. What was there pre-sex? What did microorganisms do? Yeah, they just split in half and make a clone of yourself. But eventually sex was invented, and once it was invented, it became kind of the dominant thing. So that's interesting. Photosynthesis around November 12th, and eukaryotes started to show up on November 15th. Okay, let's look at December. This is December on our galactic year, cosmic year. Yeah, we didn't even show up until New Year's Eve. So, December 1st, we started to have an oxygen atmosphere, so there was enough photosynthesis that oxygen invaded the atmosphere. And that was pretty crappy for everything until they discovered how to use it, and then it was pretty good for everything. We had like weeks where stuff was all just like unicellular organisms. There was no multicellular organisms until the 16th of December. Then we started to get multicellular organisms. We got some worms. That was pretty good. We got a lot of fishy things. We got the first vertebrates on the 19th of December. What is a vertebrate? It's got a backbone like us. So that was good, first fish. We started having vascular plants, which are more complex plants, not quite so simple. Insects, amphibians, got some trees, got some reptiles, and then December 24th we got dinosaurs. So that was pretty cool. That was a good day. Nothing on Christmas. We showed up a little bit after Christmas, first mammals. But this was still the age of dinosaurs. Dinosaurs lived and ruled for a good 4 days before they went extinct, which is pretty long compared to us. We've only, like, mammals haven't been around for that long, and humans definitely, we're not even close to the length of time that dinosaurs ruled. We kind of think about dinosaurs as like existing 65 million years ago, but that's not really true. They existed for a really long time. It's like they existed for 65 million years, 65 million years ago. So they were pretty good. We got whales and primates on the 29th. After the dinosaurs became extinct, it like opened up all of this space for new life forms to like take up. If that makes sense? Like all of these new opportunities came about.
When we had the oxygen graph, there was like some, I don't know what you said, but there was a point where like a lot of new species just like starting to make.
Yeah, I think that was up here, like right before the first vertebrates. This was called the Cambrian explosion. So there was like a whole bunch of new, just like radically different designs for life. And one of them was like symmetric, bilaterally symmetrical, with a nervous cord down the middle.
And that's pretty
And that was vertebrates.
Okay.
Yeah, so we got whales and primates after the dinosaurs died because now there were all these kind of places that life could evolve into that wasn't being taken out by dinosaurs and the other things that went extinct at that point. First hominids, which is primates that walk upright on 2 feet like us. Yeah.
Wait, so like in total, like dinosaurs only existed for less than a week?
Yes. Yeah,
We haven't
in
been,
total.
we only existed for a day.
But
We've only existed for a day.
we're like a month for like 2 years.
Yeah, so if let's see, 12.5 billion is the whole time, so 1 month is about 1 billion years. Which is a long time, right? Okay, so first humans, we showed up on December 31st, and we were late to the party, so let's zoom in on December 31st, New Year's Eve. We showed up at 10:30 at night. Oh. We are so late to this party. We started using stone tools at 11. We didn't even make fire a thing until 11:46. We've got 14 minutes since the origin of fire. So Australia did a lot of cave painting in the last minute. That was pretty good. Invented agriculture in the last 40 seconds. Since the beginning of agriculture, first cities, invention of the alphabet. I think this is the alphabet in Mesopotamia, I'm pretty sure what they're talking about on that one. Birth of Buddha, 5 seconds ago. Birth of Christ, 4 seconds ago. So this is the last 2,000 years. We'll take a little. Mathematics, as we understand it, 3 seconds ago. Crusades, 2 seconds ago. This last second was a hot time for us. Renaissance in Europe. This was sort of all of the expansion of empires across the globe. Invention of science as we use it today. In the last 2 seconds. And finally, in the last second, we've developed the capacity to destroy ourselves as a species. Hopefully not the will, but we have the capacity to. And but also our first steps at exploring other worlds happened in this last second. Just an enormous explosion of human movement around the globe, which has never happened before. It's exciting and cool. So we have we're living in a really exciting time. In these last 2 seconds, just in this time, right here, last 2 seconds, we've changed the Earth enough that if we went extinct tomorrow, distant civilizations in the far, far future, several cosmic days from now, would look back and they would know that something happened here. We've made an impact on the Earth itself that will be visible in the rocks as long as those rocks exist. Just great. I don't know. It's a weird thing. It's an exciting thing. So the plastic that we make will be in the rocks forever. The atmospheric changes that we've made, those will be visible in the rocks forever. And there will also be this thing that's kind of a major bummer, which is that we're in an extinction event right now, which means that globally the rate of extinction of all species across the globe has gone way up, and that will be visible in the rocks forever as a layer where fossils radically change. And so that's weird and exciting. We live in a really really crazy and exciting time. But we're also very, very, very young and very, very, very small. So this is an image from the Voyager spacecraft. You guys know about Voyager? Yes. Yeah, it's a spacecraft, and its job is basically just to go really, 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 really far into space. It is currently the furthest away from space, us, that any human-made object has ever been, and it's getting further and further away to us. So it has solar panels, and it can still get enough energy from the sun to send back messages once in a while. But eventually it's going to stop communicating with us, but it will still be going out in space. Because there's nothing to stop it. It's like past all the obstacles now. It is
What if you get like
moving.
pulled into like an atmosphere, like a planet or something?
Well, it's actually past all the planets. It's outside the solar system, and it's in the space between stars right now. So eventually it'll get sucked in by another star. But right now it's just out in space.
Wait, so we
And
don't,
it's going really fast, and it's cool, it's exciting. It actually has evidence of humans. They like put this golden record on it that has like recordings of civilizations from around the world, just in case somebody ever found it. It's mostly poetical. I don't think it's a real, like it's actually going to be found, but it's cool. And so as it was leaving, I think it says while it was passing Saturn, Carl Sagan, who was part of the the team of scientists who made Voyager, the thing, he was like, let's turn its camera back around and take a photograph of Earth. So this is Earth from Saturn. This little dot has all of us and everyone we've ever known and cared about. And then a new picture that is from the Cassini spacecraft, which had better cameras. So this is Saturn, this is the rings of Saturn, and this is us, right here. All of us, which is pretty exciting. So we're pretty powerful on our little dot, but we only have this one little dot to live on. And all the life that I talk about in this class, it all happens on this dot because we don't know about any life anywhere else. What's that?
Those poetic histories of civilization.
We might find more. That would be really exciting.
That would be scary.
Yeah, it would be scary. Context for like fun.
Jeez.